Hello student, once again welcome to Oni's classes. Today I am going to start your second chapter of biology, that is sexual reproduction in flowering plant. So this is the first page of your NCERT book and this is the screenshot of the second page. So in this two pages, just definition of sexual reproduction and about the pre-fertilization structures are given. So the summary of these pages are: first, the definition of sexual reproduction is given in the first page. So that is given: reproduction ensure the continuity of species generation after generation as the older individuals undergo senescence and die. So what is the significance of reproduction? If come, then you have to write this point. Second important point of the page is that flowering plants show sexual mode of reproduction and bear complex reproductive unit as male and female reproductive unit along with accessory structure. It means it is said that means in the flowering plant who is showed a sexual reproduction, they have different male reproductive organ and female reproductive organ. In male reproductive organ, the male gametes is produced, and in female reproductive organ, the female gametes are produced. Flower is a modified stem which functions as reproductive organ and produces ova or pollen. So what is flower? If question come 1 mark, you have to write it is a modified stem, and that is the reproductive organ of a flowering plant, and where ova means female gametes and pollen means the male gametes are formed. A typical angiospermic flower consists of 4 whole or floral appendices. It means a flower has total 4 number of holes. What are the 4 holes of a flower? You have to write calyx, corolla, androecium. Androecium means male reproductive organ consisting of stamen and gynecium, composed of styles, ovary and stigma. Means gynecium has 3 parts. The lowermost part is known as ovary, then style and the stigma. Next, this diagram is given in your NCERT book. So here all the holes of a flower is shown. These are the style. Style means the this is the ovary. So this is the gynecium. Here the here the stalk-like portion is called style. Then this uppermost portion where the mature pollen grain come and stick by the help of pollination process is known as stigma, and the lowermost basal swollen portion is known as ovary. And these are the endoecium. Endoecium has 2 parts. The uppermost bilobe part is called anther, and the stalk-like position is called filament. And these are the petals, the second hole of flower, and these are the sepals, the first hole of the flower. Means the 4 holes are sepals, petals, then a filament and anther make the endoecium, and stigma, style and ovary make gynecium. Next, pre-fertilization structure and event. So several structural and hormonal changes led to formation and development of the floral primordium. Inflorescence is formed that bear floral buds and then flower. In flower, male means androecium and the female means gynecium, differentiated and developed in which male and female gametes are produced. Already I have told you that in androecium, male gametes means pollen is formed, and in the gynecium means female reproductive organ where the ova or egg is formed. Next, it is the third page of your NCERT book. Here, the structure of stamen, microsporogium, and pollen grain are explained here. So here, some terms, the stamen, filament, anther, bilobe, dioecious are given. And the summary, and look, this is microsporogium pollen sac, and structure of microsporogium also explained in this page. Sorry. Now, I am going to explain the summary of this page at note-wise like this. So first, the structure of stamen, microsporogium, and pollen grain. I am trying to keep the heading same with your NCERT book so that you can easily link up which point I am explaining. So stamen consists of long and cylinder stalk called the filament. Means stalk of the endoecium is called filament, and generally bilobed anther. Means at the head portion it has 2 lobe structure, that is called bilobed anther. Each lobe contains 2 theca, means dithecias, and each lobe is divided into suppose just a minute. Suppose this is the structure of an anther, this is filament, and this is a bilobed anther. And this anther means the bilobed structure of the anther is like this, and each lobe is again divided into 2 theca. It means a total mature anther has total 4 number of theca. Means one lobe has 2 theca, that's why total 2 lobes has 4 theca. The anther is 4-sided structure consisting of 4 microsporangia, 2 in each lobe. The microsporangia develop further and become pollen sac, which contain pollen grains. So look, this is the structure of anther. This is bilobe structure. This anther. This is filament. This bilobe structure. Look, this has 4 theca. This 1 theca, 2, 3, 4. Means one complete bilobe structure contain fully 4 theca, and within each theca one microsporangium is present. It means in an mature bilobe anther there are total number of 4 microsporangium is present. So question may come, a complete mature anther contain how many number of microsporangium? Then you have to write it contain total 4 number of microsporangium. Next point, microsporangium is generally surrounded by 4-layered wall, the epidermis, endothecium, middle layer, and tapetum. In the innermost layer of tapetum nourishes the developing pollen grain. So this is the structure of microsporangium. Look, this is the structure of a 4th bilobe anther, and this anther has 4 theca, 1, 2, 3, 4, and within each theca this is 1 microsporangium, 2, 3, 4. So total 4 number of microsporangium is present. And the structure of microsporangium, when we analyze this the diagram is given in your NCERT book also. So look, this microsporangium has total 4 layers. The outer single layer is called epidermis. The secondmost layer is called endothecium. Then next 2 layer is known as middle layer, and the innermost 4th layer where one nucleus is present, that is called tapetum. And in the innermost cell there are some homozygous sporogenous some homozygous tissue or cell which are compactly arranged without any intercellular space, that are known as sporogenous tissue. Means these are the sporogenous tissue. So how many layers are there? There are 4 layers. Outermost single layer is called epidermis. Second layer is called endothecium. Next 2 layer is called middle layer, and the inner 4th layer is called tapetum. And this, the function of tapetum is what? The function of tapetum is that they provide nourishment to the developing pollen grain. And this is the structure of a young microsporangium, and this is the structure of a mature microsporangium. So if question come, draw the structure of a young microsporangium, you have to draw the diagram of this one. This is young microsporangium and this is mature microsporangium. And sometimes some question come that differentiate between young microsporangium and mature microsporangium. Then you have to write what? Then you have to write in young microsporangium all the 4 layers are present. First point you remember, means this time you will get this type of question which once whether means your conception is clear or not. So if question come, differentiate between young microsporangium and mature microsporangium. So first point, what will you write? First point you have to write in the young microsporangium total 4 layers is present. Epidermis, endothecium, middle layer, and tapetum. Whereas in mature microsporangium, only epidermis and endothecium is present. This is the structure of a mature microsporangium. Here only epidermis and endothecium. Epidermis and endothecium is present. That is the first point. Second difference, what will you write? Second difference you have to write in young microsporangium, the middle layer and tapetum is present. Whereas in your mature microsporangium, the middle layer and tapetum is absorbed, and in place of that, some pollen grains are present. Means pollen grains are present only in mature microsporangium, not in young microsporangium. So that are the 2 basic difference between young microsporangium and mature microsporangium. Now go for the next point. Sporogenous tissue. It is compactly arranged homogeneous cell which are present at the center of each microsporangium when the anther is young. So already I have explained these are the sporogenous tissue. So these are the sporogenous tissue. They are present only in young microsporangium and they are compactly arranged, means within this cell, homogeneous cell, no intercellular space is present, and from this sporogenous tissue the pollen grains are produced. Go to the next important point, that is microsporogenesis. So what is microsporogenesis? Microspore means male gametes, genesis means formation, means the process of formation of male gamete is known as microsporogenesis. Micro means male gamete. The formation of microspore from a pollen mother cell, each cell of sporogenous tissue through the meiosis is called microsporogenesis. So another question may come, if question come from where pollen mother cells are formed, then you have to write when the microsporangium become mature, each sporogenous tissue, each cell of sporogenous tissue will act as a pollen mother cell. From each cell of sporogenous tissue, the pollen grain will be formed. Microspores are arranged as tetrad. As the anther mature and dehydrate, they dissociated from each other and develop into pollen grain. Pollen grain or the male gametophyte are released by license of the anther. So, just a minute. Sorry. So each cell of microspore, suppose this is the sporogenous tissue. So this sporogenous tissue when become mature, they are divided into 4 cells, 1, 2, 3, 4. Means finally this type of 4 pollen, they are arranged in 4 microspore and they are known as tetrad. And when they become mature, they will separate it from each other and they finally give rise to 4 pollen grain. It means from 1 sporogenous tissue, means 1 pollen mother cell, 4 number of pollen grain is formed. From 1 pollen mother cell, 4 pollen grains are formed. Pollen grains have the following characteristics: generally spherical, about 25 to 50 micrometer in diameter, and their diameter is 25 to 50. Mature pollen grain comprises of 2 layers. Outer hard layer exine, made up of one of the most resistant organic material, that is sporopollenin. That term is very, very important. So outer layer is made up of sporopollenin and that enable them to resist high temperature and the action of strong acid and alkali. The region in exine where sporopollenin is absent are called germ pore. It helps in the formation of pollen tube while the pollen grain germinate on stigma. And the inner thin continuous layer is known as intine, made up of cellulose and pectin. So this is the structure, or this page is from your NCERT book. This is here the structure of mature pollen grain is given. So you have to remember a pollen grain has total 2 layers. Outer layer is exine and the inner layer is intine. So exine is made up of sporopollenin and the intine is made up of cellulose and pectin. And in the exine, in particular position, in one position, the sporopollenin is absent and that particular portion is known as germ pore, through which at the time of fertilization, the germ tube or pollen tube will be formed. Then that explanation is given here. Next, go for the structure of a mature pollen grain is given in this page, and I am giving the summary here. A mature pollen grain contains 2 cells, vegetative cell or tube cell. It is larger as compared to other cell and possesses vacuolated cytoplasm, which is rich in reserve food. The nucleus is larger and irregular. So the pollen grain has 2 cells. One is vegetative cell, another is generative cell. Vegetative cell is larger, whereas the generative cell is smaller cell and usually spindle shape or spherical with dense cytoplasm and prominent nuclei. It divides mitotically to form 2 non-motile male gametes prior to release. It means in the structure of, look, this is the structure of a mature pollen grain. So this spike-like structure, this layer is called exine. So you have to write this is exine. This next layer is called intine. Look, this is intine, and this is the in the within the, and in some portion, look here, the sporulin, this axine portion is present up to here. Again, start from here. Here the axine is absent, and that portion is known as germ pore. And within the pollen grain, there is two cells. One is called tube nucleus or vegetative cell, and another is called generative cell. And this, the function of tube nucleus is provide nourishment to this generative cell, and this generative cell later divide into two male gametes who will take part in the fertilization. So this is the structure of a pollen grain. So look, here the microspore mother cell, the summary chart, you can write that this is the microspore mother cell or pollen mother cell, means this is a cell of, means suppose sporogenous tissue from pollen mother cell, this microspore first dyad, means first this cell is divided into two, and that is called microspore dyad. Then again it's divided into, and finally from microspore tetrad. I already told you that from one microspore pollen mother cell, total 4 number of microspore is produced. Then this is the microspore. Then this microspore, look here, some within the cytoplasm, some vacuoles are present, and this is cytoplasm, and this is the nucleus. Initially, within the microspore, young microspore, only the nucleus is present. Then some vacuole appear there, and due to the appearance of the vacuole, this nucleus undergoes cell division, means it take a shape of spindle shape, and finally from this spindle shape it divided into two cells. One cell is called vegetative cell and another is called generative cell. Vegetative cell is larger and which take part in the, means which provide nourishment to the male gamete, and this is the generative cell. So generative cell function is generative, it again subdivided into two cells and which are named as male gametes. So this is the explanation of the microsporogenesis, means how the pollen, means male gametes are produced within the male reproductive organ. So today in this class we have discussed about what are the different whole of flower, next what is the male reproductive organ, what is the female reproductive organ, and the process of formation of male gamete, means the microsporogenesis. So microsporogenesis means the process of formation of pollen grain from pollen mother cell. So in my next class, I will discuss about the megasporogenesis, means the process of formation of female gametophyte or process of formation of the ova. Till then, okay, thank you.